programs. Now the library is holding diploma, B.Pharmacy, M.Pharmacy, with four specializations, pharmacy, PhD, and postgraduate pharmacy courses. Library has many credentials and applications like MBA, MAC, UGC, and PCA. Library always put forward to organize educational activities like seminars, workshops, MBA programs, national and international conferences for the sake of the development of students and faculty. In this connection today, we had the opportunity to organize a research talk on recent advances of Zebrafish Models in biomedical research. On this occasion, I would like to invite our honorable principal, Dr. Padmanabhan to promote the events. Now, I would like to invite today's guest of honor, Dr. Zebrafish Models from IICT Hyderabad to promote the event. Let us begin the program with formal inauguration by lighting of lamp. I would like to invite Ms. Chimani, today's resource person, to join the session, and I invite the dignitaries for the lighting of lamp. I invite Praveen sir to join the session for lighting of lamp and Anil for the lighting of lamp. Now I invite our beloved principal, Dr. Padmanabhan to address the gathering.
Very good morning, all of you. Respected police resource person, Honorable Sumana Madam, Senior Scientist at IICT, and Film Department, SRM, who is working on the guidance, and today's program coordinator, Mr. Jyoti, Madam, as well as the coordinator of the Institution Innovation Cell, and R&D Director, Dr. Praveen, other faculty members of our institute, as well as the other participating institutes, and dear students of B.Pharm, maybe PC program also might be there. So I welcome you all for today's seminar on very interesting topic. So So actually, this Zebrafish model is a new concept in the research field and which is being widely accepted and widely used in so many research organizations. And this is first of its kind which we are organizing at our college also. And already for the last 6 months or 8 months, this Zebrafish facility is established at Hyderabad. And still we are at the learning stage, particularly our faculty. We means our faculty are at the learning stage, working on Zebrafish models. They need some more exposure to what best that model can be extended for the other research activities. So that's why they are exploring all the ways to gain some knowledge, and particularly arranged Dr. Praveen has recently attended one seminar at Chennai in, I think, SRF Institute. Earlier also we had one at Trivandrum, and now we start on calling the experts and explaining maybe the concepts involved in this Zebrafish model, so that our faculty as well as the students of particular this area, maybe our students or students of surrounding colleges also, they can benefit. I'm happy to see more student participants from Bharati College of Pharmacy, and really I appreciate your interest. And this can be, so only 2 calls are there, and maybe till afternoon it may be. So don't waste your time and try to gain as much as knowledge as possible, because you have all we want this name. There being some amount for registration, but the thing is simply coming and spending happily is not the thing. Okay, you need to gain some knowledge which is going to be useful for your future career. Okay, so I am not going to take much of your time, and on behalf of Hyderabad family, once again heartily welcome the 2 resource persons who came from IICT, Ministry of Technology, Hyderabad. And in coming days also, we are sure that such expert talks are going to be organized at our institute. Like this is a practice at our institute, and maybe next time I request the authority also to plan for the hands-on blocking model, so that it will be known for the students also. Okay, so with these few words, I thank the organizers, particularly IAC, headed by Dr. Jyoti Madam, already headed by Dr. Praveen, for organizing such a nice event at our institute, and thank you for giving this opportunity.
Thank you, sir. Thank you for your motivational words.
Permanent faculty at University of Texas. She came back to India in 2009, and she wanted to serve Mother Nation. That's the spirit. So she wanted to train students like you from all parts of the country. So she had a lot of grants, and she had already some 10 plus students who have successfully completed their PhD under guidance, and still 8 more to go. And still she is having free running projects with her. So that should be the motto. Okay, so earning money is secondary, but she wants to serve the nation. Okay, so really appreciable.
IICT. She has completed her PhD from Banaras University, Varanasi in 1997. She has more than 25 years of experience in biomedical research, including 10 long years of international experience as postdoctoral fellow in different fields of biology like physiology and neurobiology. She served as faculty in the Department of Psychiatry, University of Texas, USA. And with her outstanding credentials, she worked several hours like Ramalika Swami re-entry fellowship, self-powered fellowship in the year 2021 as outstanding women researcher in India. She has mentored 11 PhD and 47 master's students so far, published 104 publications in peer-reviewed international and national journals with highest Google Scholar citation 7,781. And currently she is recently selected as a member status in prestigious American Physiological Association in recognition of her contribution in the advancement, communication and application of physiological science and knowledge to enrich society and improve lives. Currently, she is serving in editorial board members of several reputed neuroscience journals like Franklinius in Neuroscience, Molecular Medicine, and Secular and Molecular Neurobiology. She was selected for the year's WSM, that is Women of Substance Network 2024 award in the field of science and research. The award was presented on the occasion of International Women's Day, 8th March 2020. So this is the brief introduction about Sunaj Development, and we appreciate and congratulate Dr. Sunaj Development for her efforts in the development of the world. And I invite Dr. Sunaj Development to contribute this session. Thank you. Hello. Good morning, everybody. I am very happy to be here. Heartfelt thanks goes to Tracker organizing team, especially Jyoti Madam. I am very happy to be here. Today I am going to tell something about Zebrafish Model. How it is used and how my lab is using it. So to start with, as Madam already told, that's my career. So it's a advanced career. I did zoology honors SC, then zoology SC, and then PhD from Banaras University, Varanasi. After that, I did a short postdoc in ICG, Delhi. Genetic Engineering and Biomedical Center, International Center. I worked for 1.5 years, and then I went to University of Texas, Southwestern Medical Center, US, and I stayed there for 10 years. And last 3 years I was faculty there in Department of Psychiatry, and got green card also. But I came back here because that time India government has set on international fellowship that whoever is doing good in abroad, they can come back and start their own lab so that our people can get more benefit. And that we are doing here great, but the government initiated that fellowship. So I was the first moment to get that fellowship. Ramalika Swami re-entry fellowship. Re-entry means that actually mainstream we get from here to US, and that's why they are telling that it's re-entry. After being abroad, whoever is settling there, they are fine, I guess. But if they want to come back to get benefit, to train their own people, that they should be encouraged. For that reason, I came back, and from there, I was regularly in different positions. Next, please.
Yeah, after joining in CSR laboratory in Indian Institute of Chemical Technology, India, I put it in several, served in several positions, like he was in principal scientist, and now I am I say only senior principal scientist, and my major research interest is behavioral and molecular neuroscience, neuropsychiatric and neurological disorders, neuropsychopharmacology, and serious pharmacology. These are the short things I tell you. The basic work during the period. First one is BHU. I stayed there for a long time, like BSc, MSc, PhD, all I did from there. And after that, ICTV in Delhi. After that, this is the English Officer Medical Center, University of Texas. And here I stayed for at least 10 years. And after that, I joined IIT Hyderabad. So, let's get into the main point here. That very basic biological processes are best studied in single cells because it is easier. And single cell tissue culture, plants, because they are easier to grow, and examining is also, investigation is also easier. So, in plant cell culture or animal cell culture, you can get a petri dish, you can get the result easily. Hello? Am I audible? Okay, fine. So, but in reality, what happens? Whenever you do research in create, ultimately for getting result, getting publication, it should be okay. But in reality, actually, we have to get the human ultimately. So, but in reality, it may not be the same because it is not a system. So, if you are doing one kind of cell like liver cell or whatever cancer cell, any cell, so that is effective in that cell only. You can say in one line, okay, in liver cell, it is like this. This compound is doing like this. But it is what will happen if it is goes to as medicine in your body, then it may not be true for that. So animals, that is it. Animals in research is very essential. And what happens to the living body, which is far more complex than it is in the petri dish. So various animal models that we are using, like C. elegans, C. elegans, fish, mouse, Drosophila, amphibians, rabbit, monkey. So biologists implement organisms multiple levels, cells, organs, or whatever. So all levels of investigation was required to get a full description, understanding the mechanisms. And the hypothesis and models can emerge from in vitro studies, as I told you, that in petri dish you can do, but lately you have to do it. So animal research is always a vital part in a medical understanding because it's a system. And this has saved millions of lives worldwide. So in vitro petri dish, you can just say the mechanism of that cell, and when you are going to take further step, you have to touch the animal model. The problem is not using animal models, but the use of the model. Because all the diseases cannot happen to you in one model. It is very exclusive. So biomedical research is actually mice are the most used and then guinea pig, rabbit, amphibian fish, non-human primates. These are the different. Or mouse, why it is become popular? Because genetically and genomically, human and mouse are very similar. Because of they are mammals, first thing, and another thing is they are metabolically also little bit similar than to the human. Mouse is small, making economic choice, and also breeds very well. Scientists have tremendous knowledge about mouse physiology and anatomy. They are already knowing of this. That's why also mouse is very popular. The mouse is among the first mammalian species that genes modified in molecular tool. That is also very popular. So about the brain diseases, since I am a neurobiologist, so I want to emphasize thinking. Like brain disease are two types. One is disorder type, one is psychotic type. So disorder and injuries are cerebrovascular, if some stroke is happened, or encephalitis, or meningitis, or brain tumor. This thing, something is going or something infected, something happened with that thing. That is called brain, this kind of brain disorder. Neuroscientific is you don't have any reason behind it, not very definite reason. Like Parkinson disease, Alzheimer's disease, epilepsy, multiple sclerosis. This don't have that kind of reason that this thing happened, then only that thing happened. Once you have it, then only you will know that okay, Parkinson disease happened. So in brain diseases, global burden of brain disease is very less as compared to other diseases. And global mortality also 12% only. Now, among those, cerebrovascular diseases are more, and depression and other diseases, mental disorder are very less. I mean, I'm talking about fatal cause. About the fatal cause, it is different. So you can easily question that why you should care about that. Only 12% are dying with that. So why we should not care about cancer even more? Okay, I understand that cancer is fatal, but here you can say that quality versus quantity. If you have a mental disorder or brain disorder, your life quality of life will be, you are not dying maybe, not tomorrow you are dying, but your efficiency will be that much less that you are kind of half functional or more functional. So in this way, you can say that your function is less, so quality is happening. So quality matters. I don't think anybody will wish to die and living longer without having anything. Actually, I will, especially I don't like to be like that. Because it is there. So if I am living, I should function also. I should make others also. So mental disorder characterized clinically significant disturbance, and 1 in every 8 people worldwide living mental disorder. And among mental disorder, depression anxiety tops the chart. And again, depression and anxiety that can become a cause of death. It's very clear. Maybe with the depression, you can get heart attack or like that. But other chronic kidney disease and other things you can have. So now the question is that among the models, why we are preferring today's lecture is with a fish. So why all of a sudden we start jumping into fish? Because mice is already, I told you that mice is so much well known and genetically engineered. We can do molecular activity, this and that. So why we should jump in? So why mice and rats are the common choices? But use of zebrafish is rapidly gaining. Why? Because due to many ethically budgetary concerned traditional methods, many new strategies have been adapted. And so those models are cell culture and Drosophila and agriculture, C. elegans. These three are mostly in neuroscience they are using. But I believe that zebrafish is better than these two because this is more closer to human. At least they are vertebrate. So as I explained you about the behavioral disorder, if you see like Drosophila, do you think that is a very good model of behavior? What they are doing, that can be a response, not behavior. Right? So for fish, it is kind of way ahead than the Drosophila and sea animals. So although humans may appear extremely different from zebrafish, at least 70% of human genes are found in zebrafish. That's why also it is 70% is quite good intelligent animal. So while mice are usually more similar because they are mammals, but several advantages over rodent counterparts because they are vertebrate. So Drosophila also have counterparts. One important advantage is that adults are small, prefer to house in large groups. As a result, that zebrafish store houses hundreds of thousands of litter in a state of art facility. One can grow and can do limited research. tropical fish. They are about 2.5 centimeter to 4 centimeter long. Barbel stages it is transparent and regular stripes length of the body like this. This is male, this is female. And males are slender and females are round belly. These are the developmental stages and other things I am keeping that aside because my student Chinmay is going to get more detail about it. So specific benefits also she will do, but maybe I can do that. Zebrafish is small and robust and maintain more cheaper than mouse because it can easily understand in our language facility only. We get 500 rupees or 3 to 500 rupees for one mouse, one piece of mouse. But this zebrafish we can get 20 rupees per year, right? Or even more, 20 rupees per year. So this is little cheaper. So zebrafish eggs are fertilized and developed, and this is another advantage that eggs are fertilized outside, external, so we can manage them. In mouse, we have to take care of pregnant mother, this and that. So here, after they release the eggs and spawns are released, mothers and fathers are free to get another breeding event. So we can take care of that very easily. Like that. As a vertebrate, zebrafish has same major organs and tissues as human. Specific benefits have unique ability to repair the heart muscle. Most of the organs in zebrafish can regenerate. That is also advantage of having zebrafish as a model. It doesn't go very easily. These developmental stages my student will give you more details. It's clearly visible. Just one line I can say that if you are trying some drug and that drug is fluorescent, you can easily see because in zebrafish embryos are very transparent and you can easily see the fluorescent drug where it is going. So to trace the drug where actually it is going, it is easier in zebrafish than mice because mice inside something is happening and mice cannot see it until unless you anesthetize them. And after anesthetization, then again you have to get an imager, then that is a lot of trouble and sometimes you don't even see because your drug is so small you cannot even see. It is diluted in the other organs in the mouse. And moreover it is not transparent for that reason. So these are the advantages, simpler. This is the summary. Identity, sequence, genome, optical transparency, and then the manipulation can be easier because you have zygote with you, so you can manipulate easily with the cloning and piece the whatever techniques are. So in fish brain, you can see here the brain stem, cerebellum, amygdala, all are there. Whatever the human state, human brain is having, maybe it looks like a little different, but with the PCR and gene expression, we are able to know that who is what, like which organ is which part is corresponding to which human brain. So, more or less, zebrafish are sometimes identified as alternating model, but that will be too much if you claim alternating model. We can call it complementary model because after the zebrafish, then you can easily go to further step, taking less effort because these many drugs may not be passed through this zebrafish, and one or two mechanisms will be. So zebrafish applications may be in areas such as screening, molecular modeling, and behavior research. So screening, microbe screening, behavior phenotype if you have. So you can have a 96 well plate and you can put the drug in there and then you can interactive drugs. You can record their activity and you can say that which one is positive and which one is negative. In this way you can screen some of the drugs. So the transparency of the embryo is less. So likewise, there are two types. If you have a target, then you can directly go, Or if you don't have a target, and you are trying, then also it will be to have the Zebrafish, chemical library and automatic signals and high information content, and then you can find out further bioactive compounds. And here, there is the geological name of this. This can solve this better. So then, hypothetically, if you have this polynomial, what I have told that after reading, we can collect the zygotes and we can put it in this thing and expose it to engineered nanoparticle, and then later fluorescent based microscope can tell you that whether that the embryo is morphologically similar to control or morphologically is deforming. Then right away you can check the dose or the compound which is toxic in their developmental. For forward spreading of mutant, in this way you can make zebrafish lines also. So some you can have a specific level and then non-level female. Then you have out of Mendelian law, you can have this. Then 50%, 50% you will have. Then after that, again you can screen in. So 1, 2, 3 in third dimension, you can have this kind of heterozygous and homozygous diffusion. And then you can read among these, and you can get a clear line of your genome. Reverse spreading also can be possible. Like genome interest, you can knock out or knock the whatever you do, and then chemical analysis again you have to do the 96 well-played thing, and then you can go forward. General screening you can easily do with the zebrafish embryo here, and you can see here that count the number of box pixel and how much monopod is forming or not, if there is some deformity there, like that. So in this way here, you can do the chemical screening and microscopic observation. If there is increase of something, whatever you are checking, or normal or increase, you can easily see. So these are the few reports which are very big journals. They have reported that zebrafish chemical screen identifies nerifolin. Nerifolin, a number of cardiac glycoside family. This was very popular in 2012, and a big paper came on. And next paper also came on this nerifolin. Why I am mentioning here, because in the zebrafish model only they came to like. So unlike the series of adult models, where this is the catchy thing. And do you know adult neurogenesis? Do you heard about adult neurogenesis? Usually in brain, people tell that all the cells are arrested in some gene. So, but it is not actually true, because in mammals, people came to know later that adult neurogenesis also is there in two pockets. One is subventricular zone, another one is hippocampus, subgranular zone of hippocampus. In these two areas, still stem cells are there. Whenever you need it, whenever this is there replenishing, and whenever you need it, like you have stroke or you have some injury or something, so they are going to compensate. And in stress, they are very decreased. And the neurogenesis, adult neurogenesis got decreased if you are stressed, something like that. So, but an advantage why I am telling you this is the regular one. In human, in mammals, this thing happens. But in zebrafish, there are niches of abundant throughout the gastropontine brain axis. Neurogenesis marks are many more there. We always believe that two are the more second and other. The optic pattern is the main one, as corresponding to subgranular zone and subgranular zone. But other areas are also there. So in this way, also regeneration study, you can focus more with zebrafish. Some of the reports are there that for high throughput assay, that identification, this anti-antigen system compounds in zebrafish, neurotherapeutics, larger zebrafish, behavioral research, and these are the main big papers came up with the zebrafish models. Confocal imaging with zebrafish, this in our laboratory also this thing happened. And this is the neurogenic fish, where we can schematic showing the midline crossing this axon. So in confocal microscopy, you can easily see. If you treat them with BRDU is a radio-labeled uridine complex. So wherever DNA is dividing, that means division is happening. So that will be labeled. So if you treat them with the GFP later, so wherever the new level BRDU will be recognized by GFP, then you can have this kind of problem. Neurotoxicity also can be studied with this. And this is a very interesting paper that alarm response in zebrafish, innate fear to vertebrate genetic model. This happened in maybe 12. So their whole process of an isolated olfactory rosetta 5-month-old fish is identified by zebrafish model. So here, can you see here? This one is planting. The zebrafish skin with nuclei is green, and the red one is phalloidine. Phalloidine means the housekeeping, like the tubular structure. These are the general structure. If you see, then only you can figure out that where the nuclei is green. That's why this general skin. But cortical actin is also there. So now we are now focusing about the central nervous system. Central nervous system, zebrafish and mammal shows interesting similarities. So this is the rat, this is human, and this is zebrafish. So by 1 DTF, like 1 day post fertilization, major brain subdivisions visible. By 2 to 4 DTF, different types and different subtypes of neurons also appear. And availability of all major organs within 5 days post fertilization. Zebrafish have a complex behavioral recorder. For that reason, in our lab it is most important for that reason. They are behaviorally reliable. So zebrafish, I already told that these are the things, and behavioral study how it is applicable and telling. But why behavior? Again I am telling that behavior is also not fatal. With the behavioral disorder, nobody is going to die tomorrow. But again, that quality and quantity matters here. So you don't want to live with the bad behavior, right? The disorder behavior. So for that reason, recently, like in 1950s, what happened? All classical communicable diseases were the main fatal cause. But later, in 2020, non-communicable chronic diseases was more, like cancer, heart failure, these are that. These are not infectious. Remember. So except 2020 COVID, you cannot have any example that an infectious disease can happen in such a big fatal cause. So among that, if we omit that one, so among those, mental and brain behavior disorder contribute a major chunk of among these non-communicable diseases. And chronic stressful events are in our everyday life. It appears to cause depression, anxiety, and other things. Altogether countless. So when you do the behavioral thing, always we consider the facial expression, right? If you are angry, I can see when I talk to you, I can see easily that you are angry. That is a behavioral expression, right? Or if you are sad, with my words or something, that is also you are sad. So for behavior, it is very, very essential to know the mental state of the person which I am going to treat or which I am going to assess. And for that reason, human have very nice facial expression. You can easily see he's angry, he's fearful, disgusted, he doesn't like it. So easily you can see. He doesn't say anything to you, but you can easily see in their face, surprised, happy, sad. Now the thing is, you have to have human, but ethically you cannot do experiment with human or on human. You have to have some animal model. So what happened to animal model? You cannot have any expression. Can you see if they are happy or sometimes I have experienced some kids they say that they are very well aware of their dog's expression. I can understand. Okay, that dog may be residing with you for staying with you for long, and you are known, but that dog, only that dog. You cannot do any other dog or any other cat, but they can easily understand each other. Interspecific communication is there, but interspecific, I don't think we are that much experienced to have their just like I am advocating fish research here, but still fish also have that kind of problem. They also don't have any facial expression. But I have to use fish. So what do we do? So what do we do generally? We create that kind of situation so that they can get depressed or they can have anxiety or something like that. So how we do it in our laboratory? So this is called mouse social mimicry model. Social mimicry is one big mouse can bully the other mouse. So this mimicking, usually what happens, some powerful person always keep bullying the other person. After some time, they can get depressed. So this kind of thing, overcrowding stress, fear, and restraint stress is mimicking the jail kind of fear, cold and isolation. So we can create that. And what we create, experimentally created stressful situation for mice to mimic human stressors. And this is the left one. And the right one, it is a test. So behavior, creating one thing, stressful thing is one thing, and another thing, how this happened in body, how they are responding, that is another. So here you can see that as a behavioral test, you need to do social interaction test. Just like mimicking, if you are depressed, you don't want to mimicking, mingle with your friends or something. So just like mouse also in social interaction test. Or motivation. Motivation means so how much you want to get into that mountaineering or something, that is called motivation. This kind of thing we can create in human. We can put mouse into the water and we can record how much time they are willing to. They cannot get out, but they are willing to. Like they are swimming, so that kind of time we can easily see and record and statistically prove that they are. Okay. So this is another test called sucrose preference test. So water and sucrose, how much they are preferring to sucrose, and that also can mimic like coffee and other drink we can prefer more. When we are not feeling well, then we don't even want to take that drink because it doesn't help me to be happy. And for pleasurable content also, animal also has this kind of problem. So in behavioral analysis, you have this kind of. So in depression case, this sucrose test, preference test, motivation test, this kind of test is used for depression. And anxiety test is light and dark, elevated velocities, or open field test. This kind of thing is used for mice. And for memory test, how much time we are mobile object is. So likewise, since this is lecture is for fishes, so likewise we can have in fishes also. We translated all these things in fishes. Like memory, in previous case, white face, activity rapid test, anxiety test is novel time test like that, cognitive impairment, pre-chamber choice behavior conditional test. So all these things, this is called novel time test. So particular how much time they are going up, how much time they are in and under one third level. So that can say their mental status. Since they don't have space and they cannot smile at it, that I am happy. So we have to have assessment this kind of behavior. So these are the different condition test, previous test, all these things we use in our laboratory. Correct choice or not choice, what happened to give them training, what we should do. 12 hours they should be starved first. So they are always hungry. And then after that, whatever you give, they will be basically they will be looking for it. And that time the place they will remember. If it is green or red, they will remember. And after that, if you want to give them a negative response, then you can do maybe you want to text them like that. If you can stress them, there will be a catching, and they will be feared that they don't want to be cornered. So for that reason, in this way, we can preference them. Learn to preference that what is bad and what is good. So in our laboratory, we used to put chronic unpredictable stresses, hypoxic stress. Hypoxic stress is to mimic the stroke kind of model, because in stroke always that happens in brain oxidative stress, and chronic predator stress, that is also a fear circuitry related stress. Severe stresses for CUS, severe stresses are restraint stress, chasing, alarm pheromone, predator, and dorsal body exposure. Dorsal body exposure means in that in petri dish, if you put half of the water and you put a fish on there, their upper portion is always exposed to air and they don't like it because they always need to be immersed. So they always will be stressful to get the weightness of theirs. They will be always moving here and there so that their dorsal body will be under the water. So that is second stress. So these are the severe stressors and these are the mild stressors. So we combine them. At a day, in one day, we should give two stress, but one is severe, one is minor. Two minor doesn't help. We already standardized. Two minor in one day doesn't help, and two severe will be bad. So here you can see the isolation stress. And another thing, the most important thing, that they should not be stressed in front of others. Like if you imagine, if you are sad and if you can talk to other people, then stress will be little dispersed, right? We don't want that, because otherwise again another year will get. So this is very controlled kind of experiment. We should make them stressful on their own. Like except overcrowding stress, we should parameters assessed to confirm the particular mood disorder. Behavior parameter, novel time test, story condition, and scope test. Scope test is like a blood test. And story condition means fish always group together, right? Like animals, in human also, we always want to be in group, not isolation. If somebody is isolation, that means something is wrong, right? Days and years if they want to be isolated, that means something is wrong. And human are social animals. So in this social animal, soul condition means still, in soul condition means being put together. Always they are coming together. So if they are depressed, they always, that coming togetherness will be faster than the normal. Because in normal, you still explore. Fish still explore here and there by alone. But if they are stressful situation, they pleasurable, how is their preference for pleasurable liquid like tea paper, tea, or some solution, sugar solution. So that, but in fish, they are not having any swill tube, so they always, we always use a preferable palatable food like artemisium. Artemisium is their palatable food. So after this nutrition, we want to see that how much time they are taking to get into that artemisium and the palatable food. So that will give you the idea how much mentally they are stressed. Molecular parameters also we after we take all these things, behavioral phenotype, then we can get the brain connected and mRNA expression, protein expression, or here we will immunohistochemistry just like I explained that how much neurogenesis is going on. This kind of thing, this marker protein marker gene you can study in PCR and you can find out that whether they are replaced or not. Or after that you can treat with some potential, some compound, and whether this is rising or not. You can. So these are the novel drug tests in our lab we are using. This is exactly measurement, that's why and after that we do body cortisol because cortisol is the stress hormone. So we do it now if you are expecting some stress in your brain. So this is a proof. This ability novel time test, division in zone like control and stress. So how many times they are up? So control, they are significantly down in stress, and see down, significantly down in stress. This is very high. And latency to upper zone means the first very much first time, how much time they are, how many seconds they are in the upper zone. So this is also very high in stress. They are stress fish because they take long time to go to up. That means they are late. These are few of the freezing down experiments. Sometimes, if in video we sometimes see that it is looks like a photograph. They are not even moving, so they are freezing. That is called freezing down. So they just frozen over there. So that much time. How much time they are frozen out of this 5 minutes or 10 minutes? How much will you do? 5 minutes. Test. So after 5 minutes, how many seconds, how many minutes they are in freezing condition? Whether it is significantly different than the control who doesn't have any stress, non-stress control. It can be assessed that how much they are, what is their mental state. Single. Testing time always single. Yeah, they should not see others. Then they will be able to group by others test. And in stress condition also should be stressed separately except overcrowding. Except overcrowding because that will inside the test stress several things you can. I will show that video last. So this is soil option. As I told you, that soil option. This is together. Controls are less together because they are normal. So their mental state is good to explore others. So even their stress, when they are stressed, they will be together very easily. Okay, let's fight together like that. Better see you together, it will be less. It is more in. So now one more thing major thing I want to point out here that people may be more world maybe up to now just a decade old these things are happening, and my lab majorly works on that in brain disorder always it is sex cause. You can see here Alzheimer's disease is more in female, Parkinson more in male. Likewise, there are strictly some biasness is there. So in this way, you can easily understand that brain circuitry is different in both male and female. easily. So you should not generalize by doing only male model, and you cannot generalize that okay, this medicine is okay for male, so it should be okay for female, because it is not. Because in the see in public, several of my papers I am published that it is the even if nutrition is happening, but the mechanism is completely different in male and female. So we are actually missing the target. So this need to be attention, pay attention. So and after that, this NIH also takes steps to address sex difference in clinical research now. So without having any clinical trial in female, they are not going to approve any drug. So in that way also, at least brain research, you should consider both. So in this way also, why I am telling here, because in Zebrafish research also important for that thing, because Zebrafish can be easily identified in and sex difference, general sex difference, we have found women in this kind of cases, and women outcome. So can you find out what kind of thing are there? Like, for example, one female student can easily tell that yesterday what kind of color shirt the other people wore. Remember, we can easily say that okay, that time when I spoke with you, we were wearing that kind of thing, and I don't believe. I mean, I'm not telling all generalized meaning, but most of the men think that work is more important. Like what you were talking was more important. What kind of shirt you were talking, maybe that is not memorizing that. So what I am trying to say here, brain is subtlety is different. So fineness mostly recognized by female, but the target-oriented thing and other things mostly done by males. So in this way, I'm not telling who is inferior and superior. They are different. We should accept that they are different. So since they are different, so we should not generalize all the things together. That means something is differently going on in their brain. So same thing is happening in disease mechanism also. So sex differentiation in zebrafish or mystery unsolved. Why? Because in mouse and human, we have, if you remember, we have sex chromosomes, XY. But in zebrafish, we don't have that. But for sex difference, the advantage, as I mentioned, sex difference, rodents are especially rat mice, they have sex chromosomes. But in zebrafish, they are having 25 pairs of chromosomes, and their sex alleles, you understood allele, they are not restricted to one chromosome. They are spread in 4 chromosomes. Different sex alleles people have identified, and once that is done, once that is fixed, they cannot reverse it back. Like crocodile and sea animals, they can reverse it back. In temperature, if you increase the temperature, sometimes they become male, something like that. In crocodile it happens, reptiles, all the time it happens. Lizard, garden lizards, all of a sudden you can see all the populations are male because in the same environment. But in zebrafish, at least that is not there. Once it is female, it cannot revert back. So that means also that proves that we can easily use sex difference studies. 50 chromosomes as a 25 chromosomes in they have sex chromosomes, and they identify 2 sites, 5 and 16 chromosomes, and 2 more sites also they identify, chromosome 6 and 5 and 16. Few alleles are sex specific. So we have sex in zebrafish is a combination of combinatorial effects, genome, germ cell, and environment. But once it is done, it cannot be removed. So we continued our stress testing studies using other two stress diagrams. So these are the models and regular basis in our lab which is going on. In vivo study, mood related disorder, chronic restlessness, disorder, where restraint stress. As I told you that we are limiting that kind of stressors. And chronic social related stress is one big mouse. This mouse is moving the small mouse for 10 days, then it will get depressed. And this is chronic variable unpredictable stress, just like zebrafish in mouse also we do like that. Two stressor in one day, unpredictable way, and after 21 days they will get depressed. And these are the behavior tests we do. After paradigm, stress paradigm, we do elevated plus means, then open fit test, superspectral test, this is post-fit test, and this is social interaction. And we establish various kinds of behavioral practice in Zebrafish. Similarly, in stroke related thing also we establish stroke models, BCCO, ICO, and UCCO. These three models are there. So after that, when neurological deficit score, we can have it in Rotarod or grip strength meter and mobile object recognition test. These are the behavioral tests. Mobile object recognition test, and this is the water Morris water test. And likewise, in Zebrafish also, instead of Morris water test, they have T-maze test. And many other interaction tests, mirror bite test, or all other tests. So this is the major achievements in my tenure in IICD, that we developed mouse behavioral full facility and Zebrafish also. So I can show you the video. So how it is male to male and male to female interactions are different, I can show you. What happened? The minimizer to the male. Here's the bullying stuff. That 5 minutes they will bully. But this is male to male. Okay, subject male is CP7 and aggressor male is white. And we make them aggressor. And I tell you later maybe to kill somebody. So we make them aggressor. Not all of them are aggressor. It's 3. But the problem is, if I say the sex difference thing, can you show the female interaction? So here, the most aggressive male, if I put it in the subject mice female, they don't bully. So at least one thing is sure, the animal world, domestic violence is not there. So this cannot be modern. This cannot be modern. So I have opted other. I recently I am ready to publish. That's why I did not show that. But female aggression, generally female aggression doesn't happen. But I made them aggressive with some things. Maybe next time when you go there or you can invite me, then I will show that how we make it. After publication, only I can disclose that. So actually that is real for animal world. Female aggression is not. Until unless you do their bigger size and you are harming them, something like that. Otherwise, they are not ready to be. And female aggression does nothing. But now I have that model. I made aggressor female and he is really willing. So more than me, but the secret I am not telling you after publication. Ah. Last. The Zebrafish facility I am talking about. I have mice experience, that's why I could translate this to mouse behavior in the and this is a Zebrafish and Jimmy is working on it. And here, this kind of seamless video you can show but now body comfortable space now. This is dorsal body exposure. See, they want to get into the deep there. But in this much time, 5 minutes, 5 minutes will be there like that. That time also they are. This is isolation. One fish for 1 hour. They will be sitting alone. Why this? Yeah. This is the heat. Heat and heat of what? Cold. Because ice are there, ice. So 2 degree less. Restraint stress. So the restraint stress. So this tube, 2 mm tube is totally gold, so that water can easily transferred. Net with 2 nets, we can figure it out how to close it, and fish will be there. Fish doesn't have any, physical any stress, but they have mental stress that they cannot flow by their own. So for how many minutes will do? 1 hour. That's very stressful. That is one of the serious stress. So this is alarm thermal, right? See, this is alarm thermal. So in alarm thermal, let's talk. In alarm thermal, what happened? That in other zebrafish, you can cut a little bit, and then with that fluid, you can have a drop here. You can drop here, so that they will pheromonally aware of that. So they will pheromonally aware of that, and they can easily understand something bad happens because that tissue will drop. So they will be here and there. So sure. Okay, the other. This is predator stress. So this chickling is actually want to feed on that. But 1 minute we will be watching them, otherwise it will be difficult. So sometimes they grab their feed at least. But 1 minute, more than 1 minute we cannot stop. So in this way we can stress them, and the rest of the day they will be sitting there with the net on, so that the family can be stressful again, but they cannot harm anyone because we are not there. They might have just feed them. So that is not our aim. And here this is called tank change. In small time period, like within 5 minutes, 6 times we will tank change. This kind of stress mimicking human, that is some transferable job. Very small duration transfer, again gets settled in that kind of stress. This is again the heat stress. 2 degrees we can maintain. I mean, not too much, like 22 to 25, right? Okay, up to 30. Okay, fine. Next is. No, not that one. First, you show the how to do hypoxia is mimicking our stroke condition, where brain is not getting oxygen due to heart attack or something. Something is blocking. So how do we do that? Because in water only oxygen is dissolved, right? So we forcefully introduce nitrogen, and nitrogen is better compatible than nitrogen in water. So it will forcefully drag the oxygen out. So in this way, the water will be more nitrogen and less oxygen. So in this way, we can get that. After that, whenever we get that bound, that they are freezing down and they are sinking, we just tap them out. And this is the regular water. Regular water. That means this is called in our BCCO, that decomposition cell. So in this way, first they will just stay. They are not moving there. After 2-3 minutes, this also I published a paper. You can see that in females are recovering faster, and males are 2 minutes later. So in fish life, 2 minutes is very big.
How to know this hypoxia to cause damage?
Well, I see that it is sitting down just no, I mean we have a measurement. What? 0.6. What? Oxygen level. Then only this is happening. So more or less they are all responsible. So whenever that thing, so we don't wait for us. Just let this go happen and then we just take it. Because other two see, this is happening. After that, so after that, some of the things are happening. So the moment they always want to get up so that oxygen level can go. But oxygen is not there. So there will be standstill. This is not for the purpose of video. So if decomposition is there, they are having like this. Their movement is there, but they cannot move. That is total. That is total. So after some time, they will be again feeling movement. So I am talking about this one. Females are recovering faster. And we map the gene also, like epigenetic tree also, why they are doing that. Now they are active. After 3 minutes.
So what is the experience they are active? So how about we are in this we are
in this information is being they are recovered. Now yeah, after the recovery. Otherwise you will properly. And this hypoxia is creating cognitive problem. So one video, last video is there. That is, as I told you, before their stroke, just before stroke, we get them trained that what is good and what is bad. That memory. Like as I told you, that we are starved first, and then they will get full if they get into the green zone. We put a green zone. The outside we put it into green so that we can do the zinc. So whenever they will go here, they will get first food. So they will do that one. And another day, maybe same day or another day. Another day. No, the negative feeling, negative feeling.
3 days.
Okay.
So we need each
In red zone and green zone. In red zone, that is that thing that we are catching in the so that will remember. They will remember as they feel, and this they will remember as well. So in this, why we do it before stroke? somewhere in the control room, which doesn't have that stove, right? So for that, training would be all of them same. And then some of them we will do stove that I was here, and then testing for both of those. So see. So whenever they, okay, it is great. It is not safe. So they will always go to right. So how much time they are in green, how much time they are in red, we always count. And we found that we control, since they are not exposed to stress, so they always will be in that reward area. They are not going to come there. Sometimes we go, oh, this is wrong. Just they went to there. But for stroke people, they are getting less responsive because they have that circuitry problem now. But what was there? That learning, I'm going to remember. Like. So in this way, if your model is ready, then you can test any enhance of memory and or something, how much time there. So this will be a good one. Yeah. So these are the publications I have. Only in I have broken publication, but this is specifically for Zebrafish Models. Specifically for Zebrafish Models. If you go to those, then you can easily see the other. If you want any confusion, if you have any confusion, you can write to me. We will explain what exactly we do. So this is regarding principally like DLT means because faster what is the easily regulation etc. Third one is the drug screening or some of the drug has been so we up to end we do that some analytical some since our students is clinical institute so they are good friends so sometimes fix so we get maybe they can take it further. I don't have anything to say like that. So I have two of that kind of credits. So which are good. These are the two of the video. I don't know what is written there, but I know I'm missing it. Correct. For Zebrafish, they do different. This is the I can but the first time in anxiety model that time in our own game and after that we moved to several papers for other models. My students are my peers or my work. I always work with students and they are also me, so I am lucky to be enthusiastic students. CCB director of the building because the Zebrafish stock we got from there. So, and he is my husband, CCB. He also worked with many intellectual funding opportunity. As I told you, from the beginning only I don't have any funding problem. Whatever I write, at least 2 grants always runs in that. So there is no problem. This is about power grant, that is a very special grant for national global researchers. And last but not the least, I want to acknowledge our animal resource who sacrificed for our research. Thank you.
Thank you so much for inviting the knowledge on Zebrafish research models. As a token of our gratitude, we would like to speak and I invite Dr. B. Pradeep Kumar and Dr. Sudev to join for the participation.
Any questions? Any questions?
Mouse, Zebrafish.
Hello.
Any questions?
So you said you are going to do studies separately on the males and separately on the females because to identify the sex differences. So throughout the experiment, are we going to house the males separately and females separately?
Make separate, female separate. Because making will happen and that will give you a very good, so we should not do that. Make some female separate.
And every time we do the test, so we have to be doing only one fish at a time.
Yeah, one fish or mice. Because
That will be available.
Any questions? Is there any limitations in Zebrafish research? Limitations? Expression.
So, behavior in house is more reliable, but expensive. That's why it is a very good model to screen any anything else.
During stroke, can we identify the infraction size of brain infraction size during the stroke process?
Infarction.
Yeah, infraction.
So I tried that. Actually, what happened? For focal infarction, you can easily see the mitochondrial inactivity, so that white things will happen. But in my paper it is there, TBC is very limited. So overall it is very so directly you cannot see, but if you can see the compared with the control one, you can easily see it is lighter than control. Because this remember, this is not focal, this is global. Like this is global. So whole brain
PTC is right. The PTC is right option for right.
PTC, either you do PTC staining or you can do, I can tell you the protocol of PTC enzyme staining. So you will get reading. And in my paper it is there. So in mouse also we are doing like that. Because infarction is very difficult to see. Because that much effect will be there only in the cell. But enzyme wise, it will be same.
The sharp control is required. Sharp control is required.
Yeah, every time someone is
Sharp control. So what we will do is sharp control. Suppose BCC just up.
PCC in Zebrafish.
And in case of Zebrafish,
You don't do it. You just happily
there is no sharp tool.
some you will see or in mice, but about mice.
Okay. Thank you.
University Odisha and MD in Life Sciences from Zebrafish University Odisha, and she has qualified prestigious national licenses like CSIR Engineering, JRF 2020, AXL in 2021 to pursue her doctoral studies. Currently, she is engaged in her PhD in molecular biology under supervision of Dr. Sumana Chakravarti, Senior Principal Scientist, CSIR. Her research aims to uncover the sex-specific molecular mechanisms in information-induced cognitive impairment using zebrafish and mouse models. She has more than 4 years of biomedical research experience handling various in vitro and medical. With this great introduction, I would like to invite Ms. Chinmay to participate in the session.
Hello everyone, I am Chinmay Tripathi. I am working as a PhD scholar under the supervision of Dr. Sumana Chakravarti, Applied Biology, CSR IIT. First of all, I would like to thank Ranganathan Institute of Pharmaceutical Education and Research team, and my special thanks to my supervisor and Jyoti ma'am for giving me such an opportunity to give a brief idea regarding my work. Zebrafish maintenance and breeding. Actually, my ma'am has already discussed everything the research related to zebrafish, what we are doing in our lab. Now the basic part is, before doing research, we have to focus the maintenance and breeding of zebrafish. So let's start. First of all, zebrafish. Why is it so? They have 5 uniform pigmented and horizontal blue stripes like zebra. That's why it's named so. It's zebrafish, and the scientific name is Denio. It belongs to the family Cyprinidae, and the phylum Pharata, class Actinopterygium. It is a tropical freshwater fish. It is laterally compressed with its mouth directed upwards. This is the brief introduction. Ma'am already talked about it, but this is the brief idea regarding zebrafish. Then, before why we are standing here, just to tell why we are using zebrafish as a model organism for biomedical research. Before that, we should know what and how it comes, how it started, the journey, how it started. So we should know that this passed in 1972 at the University of Oregon. Dr. Singh develops zebrafish as a model organism for their research, developmental genetic research. Then in 1975, on 22nd July 1975, one space station Salyut 5 was launched. Here, Zebrafish was the first one of the crew members. In 1987, first transgenic zebrafish model was developed for various cancer-related research. In 1990 to 95, here you can see. Pollard, who first the transgenic mutagenesis in zebrafish was conducted by Christine Pollard in Oxford University. For just to identify the developmental mutations, and along with that, many of assays was done using, for example, first hospital assay was done using in 1995 using zebrafish model, and other than that, cancer research in now, its various mood disorders are also scientists are performing using zebrafish model. And before using model organism, we should know how much gene and genetic structures are
showing resemblance to the human. So in 2013, the full genome sequence was done in Zebrafish in Sandel Institute, where it was found that 70% of human, 70% of protein coding human genes shows resemblance with zebrafish, and out of them, 84% human disease genes shows resemblance to zebrafish. So for that, yeah, and for not for that, before for taking any animal as a model organism, we should consider three R concepts. One is replace, reduce, and refine. Reduce means we have to select certain technique just to reduce the number, just to replace the number of minimize the number of animals. Means if somebody is using mice, there will be many ethical issues. So just to constrain the ethical issues, we can take simple vertebrates for taking as a model element. Then reduce. We can reduce. Scientists should consider this fact to reduce the number of animals, but maximizing the efficiency. And refine. Refine means the animals should, all these stressful conditions should be refined while research. While doing research, all the stressful conditions when animals are used should be reduced. And why zebrafish are taking as a model organism? There are many advantages. Out of them, one is first one I already told about the genome sequencing. Why we are taking at genetic level 70% human gene and 84% of human genes like that. And besides that, external development, external and rapid development, short reproductive cycle, and this embryos are transparent in vivo imaging. So we can use in different fields. If we are taking any drug just to see how it is performing, how it is going through the whole body, we can use zebrafish embryo as it is transparent. But in vivo imaging, there are so many advantages taking this. And zebrafish in biomedical research. Biomedical research, if you will see this graph, then per year, according to government publication, per year is still increasing. If you search only keyword, zebrafish and biomedical research, you will find so many papers where in cardio, cancer research, in cardiomyopathy, and in mental disorder research. For example, cancer. If you will consider heart regeneration, for example, when people suffering from heart attack, heart stroke, then some scar happens in heart muscle. It means regeneration of muscle something. But the zebrafish has the tendency to regenerate these heart muscles. So we can, scientists can identify what are the following specific molecular techniques, molecular mechanisms responsible for regeneration of heart. If we target this pathway, then we can help the sufferers who are suffering from heart attack or heart failure. And in mental disorders also, our work is on molecular neurobiology, so I can give one example. Mental fish are social animals. They show schooling behavior. I have already shown one figure, soul cohesion, where they used to stay in groups and they show the behavior. If we will consider mental behavior, mental disorders related experiment, then they will show the behaviors that will show resemblance to human condition. If they are depressed, they will behave like depressed. If they are normal, they will explore many things like us. So we are taking zebrafish for different purposes. cell phone, we use cell phone, yeah, that one. This is why it is going here so much transparent. You should see every year also you can see the separation. If you mark with any green fluorescent protein, if you tag any drug with green fluorescent protein, you can see where the circulation, where the drug is going exactly. Actually, it's not that much clear because of cell phone. This development is so fast. After 24 hours of post-fertilization, it almost develops to their neural tube, cardiac muscle, circulation, everything develops after 24 hours of fertilization. And by going through different stages, gastrula, everybody knows gastrula, gastrula, and it will become the cattle-like structure like amphibian. And here, if you see, just look at this video. Here, the detailed development you can see from after 30 minutes of fertilization, how it is going on. Gradually the upset, the content of the upset is gradually declining. The volume of the upset is gradually declining because they don't take any nutrition from outside after 5 days. So they are totally dependent on their upset for their nutrition. If you consider diet and feeding, Zebrafish is omnivorous. Their natural diet is zooplankton, insects, filamentous algae, muscular plant mammals, etc. But now I told Lamprova are capable of independent feeding for up to 5 days and 6 days because of presence of X economy. Then after 5 to 15 days post-fertilization, you can feed commercially available 1 feed, 2 city feed. They are actually rich in proteins and lipids. They are in pellet form. Their size is 5 to 30 micron, and this 2 CD is 30 to 90 micron in size. You can feed them up to 50 ppm. Means post fertilization. And after 15 ppm onward, you can give one live artemia. Artemia means it is marine state. We used to culture it in our laboratory, and because they used to live feeds, so we prefer live artemia to feed them. Along with that, we feed them on 150 micron and 300 micron like that. And then here you can see live artemia. Live artemia picture. This is live artemia. After culture, this is live artemia what we are giving for their feeding. And this is the artemia cyst. We prefer inbred only for artemia cyst just culture. And this is the basic difference actually. Before doing any research, we should know the difference between male and female. So first one is body set. You will see male one is more slender than female. Male one is more slender than female.
females have this rounded belly structure, and men have on straight streamlined body. And the coloration you can see males are little brighter. The streaks are little brighter than female. And skin size will say every time these anal fins are larger than males have larger fins than female. For doing good research, you have to maintain carefully, you have to maintain, breed, zebrafish carefully, because we just to get healthy offspring. It shouldn't be any inbreed depression. So, maintenance, 4 steps should be considered. One is maintenance, second is breeding, third one offspring selection and rearing, then fourth one is replacement from older fish to new fish, new generation fish. So, first comes to maintenance. For zebrafish maintenance, we need to take care of several parameters. First one is housing system. When you are keeping in any tank, it should be always in recirculatory water system. That means aeration should be proper. Every time aeration should be proper, and the water parameter like pH 6.8 to 7.5 should be maintained. If you will lower the pH, the temperature, light condition, it will alter, it will impact on zebrafish. It will cause the stress condition. Then temperature, standard temperature is like 26 to 28.5 degrees centigrade. You can increase the temperature for embryo up to 120 hours post fertilization. Then standard is dark and light cycle is 14 hour light and 10 hours dark. Then environment enrichment, as I already told, they are social animals. You shouldn't keep zebrafish in their tank individually. They should be placed in group. Then feeding is also one of the parameters where you have to feed them 2 to 3 times. One time live feed and another time dry feeds. If you are giving 3 times per day, then 2 times give live artemia and 1 time pellets. Then sanitation, equipment and hygiene. It's more important. Hygiene, you should clean the fish room properly, mop with water, and don't use any soap solution or chemical while cleaning the tank. It will you can use hand scrubber just to avoid cross-contamination and zoonosis. And euthanasia means if you are finding any dead or diseased fish in the tank where all fishes are living, you should discard these fishes as soon as possible. If it is not possible to discard, there is one way where you can take ice water and keep it there for some time. This diseased fish they will die. Here you can see. This is larvae. This is after 5 days actually. This is one of the breeding culture horticulture also. Here we are culturing brine snakes, and you can these are the artemia actually. Artemia culture. You can feed them manually using rubber. Yeah, you can feed them by automated feeding machine. And clean water. You should clean water properly. If you don't have this type of sophisticated machine, you can use RO water for there. And before keeping fish, you should check the pH, temperature, breeding generally occurs in 3 steps. First, you have to choose the healthy mating pair. Male and female should be healthy, disease-free. So, under lab condition, zebrafish can breed year-round, even spawn every 2 to 3 days, producing around 100 transparent eggs per spawn. And the eggs are 1 to 1.5 mm in diameter. You can use one male for breeding tank. I'll show you later. In breeding tank, you can keep one male and one female either, or you can keep one male and 2 females. Then after selection, this is the first step, selection. What I said, you have to take healthy zebrafish from your reliable source. After 5 months, zebrafish becomes reproductively active. So 5 to 8, it should not be more than 18 months, so that their fertility will be lower. So sexual maturity region 10 to 12 weeks. Then acclimate them at least 1 month prior to breeding and maintain water temperature and diet, nutritious pellets and light food like actinia. This is the breeding tank. Breeding tank allow male and female, one male and one female for 20 minutes. Allow 20 minutes for mating. Then collect eggs using strainer and stream one video how to collect the eggs after mating. Then eggs we can't keep in RO water because they don't need external source of food. Only they need the proper environment. So we used to prepare embryonic medium according to Cold Spring Harbor protocol. We used to prepare embryonic medium, E3 medium, which is composed of different salt solutions like NaCl, KCl, CaCl2, and magnesium chloride like that at different proportions. And here is the mating protocol. The condition is if you are giving proper diet and food and maintenance, they will grow faster and you can see the difference. If you are not providing them proper diet and maintenance, they may not grow.
So what is the maturity age is after 3 months only?
After 3 months, but generally scientists prefer 6 months for any experiment so that they will become sexually mature.
We can perform only after 6 months.
Yeah, it's better. But your experiments should be conducted before 18 months because the fecundity in female will be lower after 18 months. But they will live for, if laboratory condition, they will live for 3 to 5 years. Yes. Any question? Thank you.
Thank you so much. Now I request Sudhir sir and Praveen sir for the participation of our resource person, Ms. Chinmay. I invite Chinmay to come to the station. I invite Maria Madam to join the participation. Please. And faculty having any doubts, you should contact Praveen sir, and also the madam has given some resources for that, and you can contact. And if you want to give any feedback on this session, you are most invited. Faculty or students, if you need to give any feedback, you are most invited. Thank you so much. And students are requested to sign in the registration place on the papers which are given in your details. Please check your details once again, and you can send to the people only that e-copy of the certificate will be sent to email. Thank you. I would like to acknowledge my thanks on behalf of Cyber Magazine for giving the support to organize this session, and I would like to appreciate and also convey my sincere thanks to Dr. Sumana Chakravarti Madam for sparing her valuable time with us and for the nice contribution in enlightening the knowledge of Zebrafish Models in biomedical research. And I would also like to thank Ms. Chimari Tripati for taking the session and contribution of knowledge. Thank you so much, ma'am. So, now, in this time, thank you so much. And I would like to invite Praveen sir to say the